And we'll start with Jane Oliaro from the Peter Mac Institute, whose work on T-cell memory and looking at infectious diseases. And we'll start by giving Jane her certificate and then hear from her.
Okay, well, good morning, everyone, and thank you for the opportunity to speak to you today about a little bit of research that I've been doing. My name is Jane Oliaro, as you heard, and I'm from the Peter MacCallum Cancer Centre in Melbourne. Many of you will know that the Peter Mac is a combined hospital and research centre. It's dedicated to all aspects of cancer diagnosis, treatment and research. And I'm part of the immunology program at Peter Mac. And my work is centred on understanding immune cell responses to infectious disease and cancer. So to generate effective immunity, it requires a whole army of immune cells, but the cell type that I'm particularly interested in is called a T cell. Cytotoxic T cells, or killer cells, are, as the name suggests, the cells responsible for directly killing cancerous and infected cells. To become activated, T cells need to interact with another type of specialised immune cell called an antigen-presenting cell. So, for example, during a bacterial infection, these specialised antigen-presenting cells will engulf the bacteria, process it into small fragments, and then present those fragments to the T cell. Now, this intimate communication between these two cells is critical for T cell activation. And once a T cell is activated, it will multiply to produce a 10,000-fold increase in the numbers of T cells. And each of these activated killer T cells are specifically designed to recognise any cell infected with that bacteria. So these cells are destined to die following clearance of the infection, but what remains is a pool of memory cells in the body, and they can stay in the body for years. These memory cells are poised to mount a very fast, rapid immune response following second challenge with this particular bacteria. So although a lot is known about memory cells and the characteristics of these cells, and indeed killer cells, how they arise from a single naive T cell is still not very clear. So to address this, we decided to go back and investigate this initial interaction between an antigen-presenting cell and a T cell at the single cell level. Now, I'm particularly interested in imaging this communication between these two cells, and it was during these studies that I discovered that T cells undergo their very first division while they're attached to an antigen-presenting cell. So this time-lapse microscopy movie illustrates this, where you can see the T cell, which was labelled in green, interacting with an antigen-presenting cell. Perhaps I can play that again. Interacting with this antigen-presenting cell here undergoes division to produce two daughter cells. Having observed this, we speculated that this division might represent an asymmetric cell division. And this is something that occurs during development but had never been shown in immune cells before. So what asymmetric cell division refers to is the unequal distribution of proteins into the daughter cell, and these proteins instruct that cell on what type of cell it should become. For example, a killer cell or a memory cell. And this theory is represented in this cartoon here. So the interaction of the T cell with the antigen-presenting cell leads to a division where proteins are separated differently into the two daughter cells. And this instructs these cells to become either an effector or a memory cell. And these microscopy images also show a T cell interacting with an antigen-presenting cell. So in the first panel, the T cell is just starting to undergo division. In the second one, it's starting to separate, and the third panel shows the daughter cells just prior to detachment. And if we use antibodies to mark proteins that we know are involved in T cell fate, we find that some of these proteins are only present in one daughter cell. So, for example, this protein here, which is marked with green fluorescence, is present only in this daughter cell, but not this daughter cell. And furthermore, if we isolate these two different daughter cell populations and we test them in a bacterial infection in mice, we find that only these daughter cells can protect against a second challenge, which implies that they act as memory T cells. Thank you. I'm nearly finished anyway. So what this data showed was that T cells can undergo asymmetric cell division, and this might be a mechanism by which they produce all the different subsets of T cells during an immune response. Along with our collaborators in the US, this work was published in Science magazine and has generated a lot of excitement in the field. It was listed in the top 10 breakthroughs of 2007. It's been cited over 240 times since then. Perhaps more importantly, it's provided the foundation for further research into the role of asymmetric cell division in T cell fate, but also investigations into how pathogens such as bacteria or virus can manipulate this process to their advantage. So finally, understanding how memory cells are developed will enable us to produce better immunisation strategies and also cancer immunotherapies. So I'd just like to end by acknowledging all the people that have mentored me and assisted me in this project, and they're listed in this slide. Thank you for your attention.